Hello friends, I will discuss about the protein structure at its secondary structural aspect. I will talk about the backbone structure, different types of secondary structure, the bonds involved to impart protein secondary structure or protein stability, protein denaturation, and methods to determine the conformation of secondary structure of protein. Let's start. So, three-dimensional structure of the protein which is biologically active is made by different covalent and non-covalent interaction like van der Waals interaction, hydrophobic interaction, ion pair interaction, etc. So, that will actually make its primary as well as the secondary structure. In case of primary structure, only covalent peptide bond is there, but in case of secondary structure, so many different structures, so many different bonds are involved. So, secondary structure is local regional folding or local backbone structure made by intra or interchain hydrogen bonding. There are so many bonds like non-covalent, van der Waals, hydrophobic interactions. So many interactions are involved here to make the secondary structure. In 1951, Linus Pauling have proposed the secondary structure alpha helix and beta sheet model. Characteristics. Let's talk about the characteristics of the secondary structure. So, secondary structure is the local structure, the regional local backbone structure made by intra or interchain hydrogen bondings. Many R groups of amino acid chains are also involved here, and we can find different types of assemblies are present here, and different types of non-covalent and covalent interactions are there. You cannot find always the ordered geometry of the secondary structure. There are some of the random and disordered structures are also present. Now there were some postulates or principle derived by Pauling by which actually the peptide bond structure was proposed. What were those postulates? He postulated some of the bond lengths and bond angles were distorted. Okay, number one. Number two, non-bonded atomic distance will not cross the van der Waals radii of the atoms itself. And in case of peptide bond, we know the C double bond and NH groups are there. They must be in the trans orientation. And there must be the presence of different types of non-covalent interactions are there. Based upon all those postulates, actually the peptide structure were characterized. So, we all know that peptide structure is made by, peptide bond is made by the dehydration reaction in between two neighboring amino acid residues. These peptide structures C double bond NH actually have some sort of electronic transition, CN bond. It is not the single bond in character. It has some double bond character. It is rigid and planar structure. Some delocalized electrons are getting transferred in between C and N. Okay. Now, the angle that is Cn in between carbon and nitrogen is 180 degree and omega, and it is in the same plane. Fine. But on either side of these plane, there are free rotations of different alpha carbon or amino acids. Now, all those free rotations are not too much flexible. They can rotate from 0 to plus 180 as well as 0 to minus 180. Now, what are those dihedral angles which actually give rise to the rotation? You can find the C alpha, right? This particular NC alpha angle is designated as phi, okay? And this COC alpha, this angle is designated as phi rotation. But all those rotations are not totally permissible depending upon the R group attached. Now, rotation flexibility can be determined by the Ramachandran plot. Now, here are the bonds and their corresponding angles involved to make the peptide structure. Now, what are they? COC alpha N is 110 degree, NCOO is 122 degree, CONH is 120 degree, C alpha CON is 117 degree, C alpha NH is 120 degree. The CC bond length is 1.54, Cn is 1.48 angstrom, and CO is 1.43 angstrom. The Cn, the peptide bond length, is actually 1.32, in between 1.49 single bond and double bond, that is 1.29. Okay, so it is in between them, that is 1.32, designated as it is not totally single bond, not totally double bond character. Here actually no eclipse structure are allowed, all are staggered structures. Ramachandran plot. Now, allowed conformations, statically allowed conformations of the tripeptide, depending upon the twist that it forms according to the formation of secondary structure. Some conformational maps are proposed because some structure are statically forbidden, okay, but some structure are statically allowed. The statically allowed structure are conformationally mapped in two-dimensional graph or plot, that is called Ramachandran. That is called Ramachandran plot, which was proposed by G.N. Ramachandran. So you can find the phi and psi angles in different axes started from 0 to plus 180 and 0 to minus 180. The 75% of the plots are physically conformationally inaccessible, but there are several, mainly 3 points are there. Here you can find 4 points which are not statically forbidden, and the secondary structures are plotted there. You can find the beta sheet allowed structure, the right-handed alpha helix structure, as well as some left-handed alpha helix structures are also allowed. And you can find some forbidden areas are near psi is equal to 0, very near about the allowed region. Okay, so some forbidden areas can be made to the allowed region by twists and all. So it is actually the Ramachandran plot. Factors. Now, to make the secondary structure or helical structure, some factors are there like pH dependence and side chain R groups. We all know in case of polylysine at pH 12 only it makes the helical structure, but at pH 7 it cannot. Why? Because at pH 7 the lysine have all positive charge, so they will repel each other, but at pH 12 the neutralized structure help to make the twisted helical structure. The side chain R group, it is very important because when I will talk about the preference of amino acids, you can find in alpha helix some structures, some amino acids are allowed and some are not. That is also applicable for the beta sheets as well. We all know the proline having bulky structure, this is not allowed in case of beta sheets as well as in case of normal alpha helical structure, because this nitrogen is not actually involved to make the peptide bond, it is within the ring, so that actually creates the problem. Next, the prediction of secondary structure. The prediction by frequency P, actually the secondary structure is predicted according to the different amino acids, because some amino acids are allowed for alpha helix and some amino acids are allowed for the beta sheet as well as beta turn. In case of beta turn, aspartic acid is allowed, glutamic acid is allowed. In case of alpha helix, you can find the presence of alanine, methionine, etc. In case of beta sheet, what are the allowed amino acids? Mainly isoleucine, valine, they are allowed. Let's see what are the causes for occurrence of all those amino acids. Suppose in case of alpha helix there is a regular hydrogen bonding, fine. So if the serine threonine is present having hydroxyl group, that will tend to form extra hydrogen bonding, so interrupt helix structure. The bulky structure of the proline inhibit the helical geometry, as well as tiny structure of glycine also inhibit the helix structure. But you can find as well as isoleucine, valine, which are applicable for beta sheet structure, is not applicable for alpha helix. Why? It will impart more hydrophobic interaction, but that is allowed for beta sheet structure. But for beta sheet structure, glycine will not be able to make the hydrophobic interaction as well as alanine, so they are forbidden. Proline is allowed to make the hinge region in case of immunoglobulin, in case of helix structure, because after some continuous structure some hinge region can be present, but it is not yet allowed for beta sheet structure. Protein stability. To give the protein secondary structure stable, to stabilize them off, there are different types of bondings are there. What are they? Hydrogen bonding. In case of alpha helix, we can find intrachain hydrogen bonding. In case of beta sheet, interchain hydrogen bonding is present, as well as the polar capping on either ends of the alpha helix. There is a polar capping made by differential hydrogen bondings, as well as hydrophobic interaction, CH2. So these actually impart the compactness of the protein because the hydrophobic residues are buried inside to avoid the thermodynamic interaction with the polar solvent. So polar structure, polar residues will protrude outside. Not only that, there are some feeble ion pair structure also, that is called as salt bridge interaction, made by NH3, extra NH3 group of lysine and extra COO minus group of aspartic or glutamic acid. That is called the salt bridge interaction. That is actually found to make the structure of hemoglobin. Apart from which, the disulfide linkages are very important, found in case of immunoglobulins as well. Disulfide structure make intrachain or interchain bonding. As well as we can find various types of van der Waal interactions, that is permanent or induced dipole. If the dipoles are permanent, that is called, as the force is called Kizom force. If it is permanent which induces another dipole, that is called Debye force. If both the dipoles are made by the induction, that is called London dispersion force. But all other forces are involved to stabilize a protein structure. Apart from which, we know the Schiff base intermediate in between NH2 and aldehyde gave by the lysine in case of tropocollagen structure. Okay, so Schiff base can be made. We can find the secondary amide bond, that is also fine in case of tropocollagen structure to bind the interchain bonding. Okay, to bind all the tropocollagen structure to make the collagen structure, as well as the aldol condensation. We know that the lysine is getting converted to L-lysine by lysyl oxidase. Okay, so all those L-lysine having C double bond O can involve to make the aldol crosslinks, to crosslink the tropocollagen interchains, tropocollagen chains. So these all are the bonds involved to make the protein structure. The protein denaturation. So if the three-dimensional structure of the protein is abolished, the function will also be abolished. The protein will be exposed together, flattened, viscous structure will be there. So the viscosity coefficient will be increased, diffusion coefficient will be increased. So that is called the denaturation of the protein, the structural and functional changes. So there are so many causes for the protein denaturation. As we know that some physical agents are there as well as the chemical agents, and if we take out the denaturing agents, then the protein will renature its original structure and function. The physical agents include heat, UV rays as well as freezing, but there are some chemical agents are also there. The agitation is also they can also cause the protein denaturation. The chemical cause includes the different types of organic solvent, okay, guanidinium ion, urea, ethanol. They can also, there are different types of organic solvents are there, chloroform, they can actually disintegrate the protein structure. Not only that, the salts like, and positive and negative ions, lithium, SCN minus, thiocyanide, there are different ions are also there, which actually, ions means they interrupt the ionic structure. The hydrophobic urea, detergent like sodium dodecyl sulfate, they interrupt the hydrophobic interactions. So that is the counterpart of the bonding. So bonding actually help to make the stability, and denaturing agents actually disrupt all those bonds, either hydrophobic interaction or hydrogen bondings, etc. Okay. So detergent like sodium dodecyl sulfate also a potent denaturing agent. Secondary structure prediction. Secondary structure can be predicted by DSSP code, dictionary for secondary structure prediction. So there are different codes are there, G for 3-10 helix, H 3-turn helix, H for 4-turn helix, I for 5-turn helix, E for extended beta sheet, C for coil, etc. There are different types of secondary structures are there. You can find from here the helical or alpha helix structure, sheet or beta sheet structure, non-repetitive structure including turns, loops, etc. As well as there are some disordered regions. First, helical structure. Helical structure means local regional folding to give the twist. Okay, so that is called the helix. So we know that in case of helix, some n number of residues are there per turn, as well as some pitch or rise is there. So distance to form a helix d is equal to p by n. As well as for the nomenclature of the helix, it is termed as n and m. Okay, so what is Nm? N is the number of residues per turn, and m is the atom. N are the atoms involved to make the helical twist, one helical twist. Let's see helix types. From here you can observe the 2.27 helix, you can observe the 3.613 or alpha helix, 310 helix, as well as pi helix. So by different types of number of residues per turn, different pitch, different rise, different types of helices can be made according to the geometry. 2.27 ribbon. This is a very constrained structure. Actually, naturally, it is not yet frequently available. Here we can find 2.2 number of residues per turn. Actually, the 7 atoms are involved. Here, the hydrogen bonds are made in between C double bond O and NH of I to I plus 2 residues. Okay, and pitch is greater than 6 angstrom. Next, alpha helix. Alpha helix is also termed as 3.613 helix because 3.6 residues per turn are found. The pitch is 5.4 angstrom, and we can find 1.5 is the axial distance. It is called also as the Pauling-Core model. And why it is 3.613? Because 3.6 residues per turn and 13 atoms are involved to make one twist, to make hydrogen bonding in between C double bond O and NH involving I to the I plus 4th residues. There are some characteristics of the alpha helix. It is found in nail, hair, alpha keratin, etc. And we actually find there are some specific helix formers are there. What are the helix former? As we know that alanine, as we know that methionine, that is very well known, phenylalanine for alpha helix. But serine, threonine, proline, glycine, as well as there are so many other amino acids, that isoleucine, valine, they are not yet helix former. They actually break the helix structure. And we can find it is made by the 5 to 53 amino acid residues. So this is the structure of alpha helix. 310 helix. It is 3 residues per turn are there, and the bonds, hydrogen bonds are made in between I to I plus 3rd residues. And in case of this 310 helix, 10 atoms are involved to make one twist. We can find actually 5.8 to 6 angstrom. This is a rise present in 3.10 helix. Pi helix, it is broadened, flattened helix because the hydrogen bond is made in between I to I plus 5th residues. It is also termed as 4.1 or 4.416 helix because 4.4 or 4.1 residues are present per turn, right, and 16 atoms are involved. Actually, these helix are too much flattened to make the van der Waals interaction, but tiny hole for the access of the water. So it is not yet actually stable. Okay, and you can find 1.5 angstrom pitch here. There are some synthetic homopolypeptides are there. So they are made by the polyproline, polyglycine, because proline twisting is not yet actually found, because proline have very bulky group and N is actually within the pyrrole ring. It is not involved to make the peptides bond, etc. Polyglycine is also made. Glycine is very tiny molecule, and this proline and glycine is actually present in collagen to give its tensile strength as well as interchain bonding, and it is actually present to give the flexibility. So coiled coil, it is dimer or trimer of the helix structure found for gene expression like transcription factor, and triple helix, that is 3 helix together, is found in case of collagen. Tropocollagens are the subunits, and it is gly-XY. These sort of repeats are there where X and Y are the proline and hydroxyproline. This hydroxyproline is needed to make the interchain hydrogen bonding. So this is the helix geometry by which you can find different amino acid residues per turn in 2.27 helix, alpha helix, 310 helix and pi helix along with its pitch. There are some rotational angle with respect to axis are there. It is around 25-27 degree in case of alpha helix, and it varies. beta sheet. It is a sheet-like structure, and beta sheet structure is actually 5 to 15 residues containing. We can find this sort of structure in silk fibroin, spider web, beta keratin, etc. And here the interchain hydrogen bonding zigzag structure is present, and interchain hydrogen bondings are made in between beta strands. On other hand, it is sometimes called a super secondary structure because 2 units of secondary structure beta strands are there. It is zigzag asbestos like structure. The distance in between 2 neighboring amino acids is 3.5 angstrom and pitch is 7 angstrom. And hydrogen bond is perpendicular to the axis of the beta sheet. Beta sheets is very much available. It is extended, fully extended structure. Isoleucine, valine make much more comfortable hydrophobic interaction for beta sheet structure, but glycine is not yet allowed, as well as proline is not yet allowed for beta sheet structure. The types, it includes parallel and anti-parallel. In case of parallel, you can find both are in the same direction from N to C and N to C, and in anti-parallel both the directions are in the opposite way, okay. But usually the parallel helices, the parallel types are not yet found because hydrogen bonding are slanted, okay, you can find here. But in case of anti-parallel beta sheet, sometimes it is coming closer, sometimes it is wider, okay. So it is easy to make, and biologically anti-parallel are much more found. Non-repetitive structures. There are so many non-repetitive secondary structures are there. Major important is reverse turn, beta turn or beta bend. It is made by the 4 amino acid residues, and it actually connects to anti-parallel beta strand. It is actually made by the 4 amino acid residues, and it may be either over the plane or down the plane, it may be possible, and it may be found over the surface of the protein. And these reverse turn or beta bends allow the aspartic acid or asparagine mainly. You can find the type 1 and type 2 beta bend according to the differential torsional angle. In case of type 2, you can find R2C double bond is outward. Here you can find the hydrogen bonding in between first and second amino acid giving rise to C double bond O. They donate it with the donation of third and fourth amino acid residue by the donation of NH. In this way, the hydrogen bonds are actually made here. Apart from which, here you can find the omega loop. The omega structure is present. This omega loop may be 2 to 15 amino acid residues. They actually over the surface of the proteins are found, and we can also found the hydrogen bonding in between the first 2 amino acids and last 2 amino acids also. Another important non-repetitive structure. So OMP-F is actually found containing the omega loop structure. Random coil means totally distorted, okay. So they may contain native structure, they may contain the disintegrated structure, okay. The sets, all sets are together. They anytime may be flipped, anytime may be twisted to any structures. So these all are the non-repetitive structures. Disordered region. So disordered region is actually we can find in case of the presence of some polar amino acids over the surface like polylysine or the presence of lysine. So it is unable to make the hydrogen bonds, it is unable to interact by making of ion pair interaction with the solution, okay, with the solvent system. So it is actually the disordered region. It can be found in snake venom as well. Methods to determine the conformational structure. So to extract the secondary structure, 2 methods are actually implemented. What are they? The ORD, optical rotatory dispersion method, as well as circular dichroism method. So they actually give you, in case of beta sheet structure, they give the poly structure. In case of alpha helix, it is dependent upon the length. So in this way, we can have the idea whether alpha helix or beta sheet is present and how far it is distorted from the standard level. So I have covered up different types of the secondary structure, their geometry, their abundance, as well as different bonds involved to make their structure, as well as for the disintegration and method to monitor, to explore, to extract their conformation. Thank you.